Hello everyone, myself Vidya Jadhav, student of Shivajirao S. Zondai College of Pharmacy, Asangaon, along with my group members Yukta Isame, Sakshi Jadhav and Sandesh Jadhav, and under the guidance of Dhananjay Kinikar sir, we did formulation and study on effectiveness of herbal hand sanitizer. After COVID-19, hand sanitizer has become an important part of our life. Basically, they are used for killing viruses, bacteria, germs. So, to study how effectively they kill the viruses, bacteria, we did a survey. We did a study on their effectiveness and formulation. Introduction. The word hygiene derived from Hygeia, the goddess of healing. Today, hygiene is associated with prevention of disease and promoting health. The importance of hygiene is usually recognized and evidence based. Physical contact between people and object is a key vehicle for the transmission of pathogens. Therefore, effective hand hygiene is a very important in disease prevention. It is estimated that simple hand washing could save 1 million lives in a year, and many public health campaigns worldwide have addressed hand hygiene with varying success. In early 2020, WHO declared a pandemic, severe acute respiratory syndrome coronavirus 2, better known as COVID-19. They are used to break the chain of infections, making them one of the important protocols for reducing the burden on healthcare. A range of hand sanitizers are available with various combinations of ingredients and mode of delivery. Given the popularity of hand sanitizers during this pandemic, it is important to understand which type of hand sanitizers work best against this novel virus. In this topic, we will discuss the role of various herbal based hand sanitizers in effective elimination of bacterial and viral pathogens with the focus on the effectiveness against enveloped viruses such as SARS-CoV-2. Definition: a hand sanitizer, according to latest FDA definition, is a supplement or alternative to hand washing with soap and water. Hand sanitizer, also known as a hand antiseptic or hand rub, is a product that is applied to the hands to remove common pathogens in the hands. Hand sanitizers are usually available as foam, gel, or liquid. Various preparations are available, including gel, foam, and liquid solution. Types of hand sanitizers. Hand sanitizers can usually be divided into two types: alcohol free hand sanitizer and alcohol based hand sanitizers. Alcohol free hand sanitizers include antiseptic, chlorhexidine, chloroxylenol, iodine, iodophores, quaternary ammonium compounds, triclosan. And alcohol based hand sanitizers contain 60 to 90% of alcohol, that is ethanol, isopropanol, and propanol, and hydrogen peroxide. Glycerin, fragrance, and colorant are used in both types of hand sanitizers. These are the different forms of hand sanitizer: gel, foam, cream, spray, and wipe. In this slide, there are the benefits of hand sanitizers. Require less time than hand washing, act quickly to kill microorganisms on hands, are more accessible than sinks, reduce bacterial count on hand, do not promote antimicrobial resistance, are less irritating to skin than soap and water. Some can even improve condition of skin. No need of water to wash hands. So these are the benefits of hand sanitizers, and the next part will be explained by Sandesh Jadhav.
Herbal hand sanitizer. This antimicrobial sanitizer contains tulsi and more other herbal essential oil as herbal constituent. which is strong natural antimicrobial agent. We enlisted top 5 herbal hand sanitizer brand in India. First, Herbal Strategy Nature Clean Hand Sanitizer. Second, Trivier Natural Alcohol Free Hand Sanitizer. Third, Palm Safe Alcohol Free Hand Sanitizer Foam. Fourth, Puro Herbal Hand Sanitizer Gel. Fifth, Herbo Clean Mint Hand Sanitizer. This sanitizer have antimicrobial properties. It is made using essential oil and is completely alcohol free. With powerful ingredients like basil, rosemary, eucalyptus, clove, neem, and of course mint. This is a sanitizer that's ruthless on germs. It's also softer on the skin. Key ingredient of herbal hand sanitizer: aloe vera, tulsi, neem, lemon, jasmine, processed in prasanna. Why we used a liquid extract of above herbs is explained in this slide. We used lemon because saponin compounds are antibacterial compounds that damage bacterial cell membranes. Besides that, lemon juice contains vitamin C, which is useful as antioxidant, and it kills molds, bacteria, germs, and healthy alternative to fragrance. Why we use jasmine? Because its results showed that the jasmine extract had an effect to inhibit the bacterial growth. It is known for its soothing, calming, and rejuvenating properties. It, it revitalizes, stimulates, and moisture to the skin. Neem. It is recognized as medicinal plant and it is well known for its antimicrobial, antibacterial, antimalarial, antiviral, and antifungal properties. Vitamins and fatty acids available in neem improve and maintain the elasticity of the skin. Aloe vera. It has potent antibacterial, antifungal, and antiviral properties to give complete protection from any viral attack. Tulsi. In modern research has revealed that tulsi has an antibacterial, antiviral, and antifungal activity that includes activity against many pathogens responsible for human infection. The further video will explained by Sakshi.
Formulated herbal hand sanitizer. The drugs that we used in these hand sanitizer is tulsi. The biological source of tulsi is the Ocimum sanctum and Ocimum bacillus. It belongs to the family Labiaceae and its uses for hypoglycemia, immunomodulator, anti-stress, analgesics and anti-inflammatory. Second one is the neem. The biological source of a neem is the Azadirachta indica. It belongs to the family Maliaceae and is used for fertilizers, pesticides, antimicrobial, anti-fertility, anti-cancer and anti-inflammatory. The third one herbal drug that we used in hand sanitizer is aloe vera. It belongs to the family Liliaceae. The biological source of aloe vera is Aloe barbadensis and its uses for wound burn healing, anti-inflammatory, antifungal, skin disease and etc. like that. Ingredients that we used in this formulation are tulsi, aloe vera, neem, rose water, glycerin, ethanol and mortar and pestle. Procedure of the formulation. Firstly, 12.5 gram of tulsi and 12.5 gram of neem leaves were dry in oven at 50 degrees Celsius and coarsely grinded to get a powder. Add this powder to a sufficient water and boil this water with the leaf powder thoroughly for at least 8 to 10 minutes. Now, add the aloe vera gel for freshly cut aloe leaf and boil water for more 5 minutes. Filter the mixture and separate the filtrate. In other flask, add 90 ml 66.5% isopropyl alcohol and 3.5% ethyl alcohol in the flask. Then in that add 2 ml glycerol and 4 ml rose water. After that, mix the filtrate and above solution completely. At last, soak the sanitizer mixture properly in the sanitized water. Evolution of hand sanitizer gels. First one is the organoleptic test. The prepared sample were inspected visually to check the texture, odor, and color of the gel in a semi-solid condition. pH evolution. The pH measurement of the formulated gel was measured using a digital pH meter. The pH measurement represents the mean plus and minus standard deviation of the three replicates. After performing the pH evolution test, the pH of our prepared hand sanitizer is 6. Gel spreadability. The spreadability of the prepared hand sanitizer was evaluated according to the methodological description as follows. Firstly, we have to take 0.5 gram of the formulated gel was sprayed on the pre-marketed transparent glass with 2 cm diameter. Then another transparent glass was placed on the top, followed by adding 5 gram. Wait for 5 minutes to disperse the content. By this method, the spreadability was measured based on the slip and drag the characteristics of the gel. An excess of the gel was scraped off from the edge and diameter of the sprayed area of the formulation was determined. The spreadability of the gel was calculated using the formula, that is equals to m into L divided by T, where S is equals to spreadability, m is the weight in the pan which is tied to the upper side, L is equals to length moved by the glass slide, and T is equals to time in second taken to separate the slide completely from each other. Next part will be explained by Yukta Isame.
Antimicrobial zone of inhibition. This is one of the tests for checking the effectiveness of hand sanitizer. In simple words, zone of inhibition is zone or area created by given antiseptic, here as we have used hand sanitizer, in sample of microorganism containing media. This zone of inhibition is having potency of antiseptic, that how much it may kill microorganism present in a given sample. Before starting with the test, I would like to share info on some terms used in following tests. First is inoculum. Inoculum is a substance used in inoculation method. It is the method to introduce microorganisms. Second term is culture media. Culture media is also called as growth media, which is used to grow some of the microorganisms in it. It contains nutrient agar, where Staphylococcus bacteria grow in this media. The evaluation test performed by us, we used Mueller-Hinton agar as a culture media, where microbe aureus growth occur well, and as well as Staphylococcus hemolyticus also grows. It is also used to isolate and maintain Neisseria species. So let's begin with our test, that is antimicrobial zone of inhibition test. To evaluate the antimicrobial activity of the prepared hand sanitizer gels, the zone of inhibition test against different gram-positive and gram-negative bacterial strains and a yeast was performed. 3 commercially available hand sanitizers were also assessed as experimental controls. A final concentration of 1 into 10 raised to 6 CFU per ml inoculum, here CFU means colony forming unit, was equally distributed on the surface of agar plate. A sterile microbiological disc was dipped into each hand sanitizer gel and allowed to dry for a few seconds, and then positioned on Mueller-Hinton agar plate. All plates were incubated overnight at 37 degrees Celsius. And then at last, the diameter of clear area of no growth around each disc was recorded. Therefore, the result represents the mean plus minus standard deviation of 3 replicates. Here of the test performed. So, next evaluation test for checking effectiveness of hand sanitizer is agar well diffusion test. It is also called as disc diffusion test. The agar well diffusion test was carried out as a preliminary screen to assess the antimicrobial activities of the various products. This involved the use of an inoculum corresponding to 0.5 McFarland. So this 0. McFarland is standard. This standard is prepared by mixing 0.05 ml of 1.175% barium chloride dihydrate, that is BaCl2, with 9.95 ml of 1% sulfuric acid, that is H2SO4. Next step, the test inoculum was swabbed, that is spread or inoculated to a Mueller-Hinton agar plate. This Mueller-Hinton agar plate contains beef extract, acid hydrolysate of casein, starch and agar. So Mueller-Hinton agar plate and allowed to stand at room temperature for 15 minutes. Following this, 4 wells were created on plates using a 6 mm cork borer. Here, in place of cork borer, we can also use backside of micropipette tip to make a well. And 0.2 ml of differing concentration, that is 100%, 50% and 25% of the test substance added to an individual well. After 24 hours in incubation at 37 degrees Celsius, the zone of inhibition were then measured. So in this slide we can see in first picture there is no hand sanitizer applied on a plate, and second picture is shown where the hand sanitizer is been applied on a culture media. So after 7 days we have observed that where the plate which doesn't contain sanitizer, there is the growth of microorganisms seen, whereas in the plate where there is a hand sanitizer, there is minimum growth occurred in microorganism. Then after 15 days, it has been observed that the plate which doesn't contains hand sanitizer has maximum growth of microorganism, and compared to that, the plate containing hand sanitizer has no or minimum growth of microorganism. Conclusion: hand sanitizer showed to be effective in limiting bacteria growth when compared to no hand sanitizer. This shows that hand sanitizer is effective in limiting the spread of bacteria. However, soap and water showed to not be as effective as hand sanitizer, which is opposite than expected. In simple language, I will explain this test. Take a petri dish, inoculate it with some bacteria or microbes. Then puncture a well using backside of micropipette tip. Remove this part of culture media using sterile needle. Then wells is been made in this plate. And remember that before making the well, we have to inoculate this media using L-shaped glass rod and spread it throughout the plate. After making the well, fill this well with melted Mueller-Hinton agar. We use this agar because it allows better diffusion of antiseptics than most of other plates, which leads to truer zone of inhibition. Then add prepared extract to well, which can hold 10 microliter of the extract, that is hand sanitizer. After adding this, Add positive control disk to other side of plate. Then incubate this plate at 35 to 37 degree overnight in upright position. After inhibition, look at the major zone and see the growth of microorganism covered in the zone containing sanitizer. Next is label. The label should contain active ingredients and the purpose of these active ingredients. Uses like hand sanitizer to help reduce bacteria that potentially can cause disease. Warnings like flammable, keep away from fire or flame, and it is used externally. When using this product, do not use it in a or near the eyes, which can cause irritation. Keep out of reach of children. If swallowed, get medical help or contact a poison control center right away. Direction: put enough product in your palm to cover hands and rub hands together briskly until dry. Children under 6 years of age should be supervised when using this product. Other information should contain storage and may discolor certain fabrics. Inactive ingredients like glycerin, hydrogen peroxide, purified water should be maintained. These are the some of the content used in label. Precautions should be taken while using hand sanitizer. Keep sanitizer out of your eyes and nose. Use hand sanitizer in a well ventilated area. Keep out of reach to children. Keep hand sanitizer away from heat and flames as it is flammable. Last is conclusion. It is concluded from various studies that herbs are very useful agents that can be used in hand sanitizer with lesser side effects and long lasting effects. Hands are the most common mode of transmission of pathogens to patients, and proper hand hygiene can prevent health care associated infections and the spread of antimicrobial resistance. Herbal sanitizer formulated by us showed to be effective in limiting bacteria growth after evaluation. This shows that hand sanitizer is effective in limiting spread of bacteria and more other microbes. Here are some of the reference which we have referred for making our project. One bottle saves hundreds of lives.
Thank you.